All right, welcome to this high-yield explainer. If you're an MBBS student gearing up for your board exams, you are in exactly the right spot. Today, we're taking down neurocysticercosis, or NCC for short. We're going to cut through all the fluff and focus purely on the heavily tested, must-know facts. We'll give you the mnemonics and clinical frameworks you actually need to absolutely ace these questions. So, grab your notes, get focused, and let's master this together.
Here's our game plan for today. We're hitting 5 main pillars: the Taenia solium life cycle, the definition and types of NCC, the clinical features, the detailed lab diagnosis, and we'll wrap it up with treatment and prevention. Let's get right into it. Starting off with Section 1, the Taenia solium life cycle. Let's look at the etiology and the fascinating, if slightly gross, journey of our pathogen.
Okay, listen up because this is a massive exam trap. For neurocysticercosis, the definitive infective stage for humans is the eggs of Taenia solium. You absolutely cannot confuse this with the larval stage. Ingesting the larvae causes intestinal taeniasis, like when you eat undercooked pork. But for neurocysticercosis, you are ingesting the eggs. Highlight this, star it, whatever you gotta do, just remember it.
So let's trace this journey. Once those eggs are ingested, the embryo, which we call the oncosphere, is released. It basically acts like a tiny drill. It penetrates the intestinal wall, hitches a ride in your portal circulation or mesenteric lymphatics, and disseminates everywhere. Subcutaneous tissue, muscle, eyes, and, yep, the brain. Fast forward 7 to 9 weeks, and it transforms into the larval stage, the Cysticercus cellulosae, depositing itself as a cyst.
Now, examiners absolutely love this next concept: auto-infection. A patient can literally infect themselves if they already have an adult tapeworm hanging out in their gut. This happens in 2 ways. External auto-infection is pretty much what it sounds like: poor hygiene where contaminated fingers transfer eggs from feces right back to the mouth. But internal auto-infection, that's wild. That happens when reverse peristaltic movements literally throw gravid, egg-filled segments back up into the stomach. Just remember, to have auto-infection, a prior episode of intestinal taeniasis is an absolute must.
Think about the mature cystic cysticercus cellulosae for a second. Physically, it's small, measuring about 5 millimeters long and maybe 8 to 10 millimeters wide. It's spherical, yellowish-white, and enclosed in this thin collagenous capsule. But the absolute highest yield anatomical feature is the inner chamber containing the growing scullex. This tiny structure is the culprit causing all that trouble once it lodges in human tissue.
Moving on to Section 2, definition and types. Let's clearly define the specific neurological infection we are studying today. I need you to lock this exact definition into your memory right now. Neurocysticercosis is a neurologic infection caused by the larval stage of the tapeworm Taenia solium. Notice the wording. The human ingests the eggs, but the actual disease in the brain is caused by the space-occupying larval stage that develops from those eggs. Don't let them trick you on that.
When NCC sets up shop in the central nervous system, it generally falls into 2 buckets. And here you've got to apply the rule of most common. Parenchymal NCC involves the brain parenchyma itself, and this is the most common site, period. Then you have extraparenchymal, which involves the meninges, the ventricles, the spinal cord, and the subarachnoid space. Knowing that parenchymal is the most common will absolutely save you on rapid-fire multiple-choice questions.
All right, Section 3, clinical features of NCC. How does this pathogen actually manifest when a patient walks into your ward?
This next part is pure gold for your exams. Listen to this. Neurocysticercosis is the most common parasitic CNS infection in humans, and literally the #1 cause of adult-onset epilepsy worldwide. Look at that first bullet, seizures. They happen in a whopping 70% of cases. It is the ultimate red flag. Now, if the cyst is extraparenchymal, the most common feature is hydrocephalus, which really carries a poor prognosis. You might also see signs of increased intracranial pressure, like headaches and vomiting, along with focal neurological deficits or even dementia. As these cysts live and eventually die inside the brain, they go through 4 very distinct morphological stages. Grab a pen and write down this memory trick right now. Very nasty neurological cysts, VNNC. That stands for vesicular, necrotic, nodular, and calcified. Examiners love to test you on this exact chronological order. Why? Because the clinical presentation changes drastically depending on whether that cyst is in the early vesicular stage or if it's in the late dead calcified stage.
Let's slide into Section 4, detailed laboratory diagnosis.
To frame our whole approach to the diagnostic workup, think of the acronym RIHFF. That's radiodiagnosis, immunodiagnosis, histopathology, FNAC, and fundoscopy.
Starting with the R in our acronym, radiodiagnosis. When you look at a classic board exam CT scan of the brain for this condition, you'll clearly see multiple ring-enhancing lesions. But the absolute dead giveaway, the thing that practically screams NCC, is that tiny hyperdense dot right inside the hypodense vesicle. That is the eccentric scolex. If you see a ring-enhancing lesion with a little dot inside it on test day, you immediately think neurocysticercosis.
Now, you'll often be asked to choose between ordering a CT and an MRI. Here's how you decide. CT scans are fantastic. Honestly, they're often better for detecting those late-stage calcified cysts. They just pop right out as bright, hyperdense dots. But an MRI is vastly superior for pretty much everything else. We're talking extraparenchymal cysts hiding out in the ventricles, subtle inflammatory changes, and early vesicular lesions.
Next up in our RIHF acronym is immunodiagnosis. ELISA is great for detecting antibodies because it has high sensitivity. But the real star of the show here is the Western blot, also known as EITB. It uses these highly specific 13 kilodalton lentil lectin purified glycoprotein antigens. Yeah, that's a mouthful. Just remember LLGP-AG. Because of this, its specificity approaches 100%. So if a question asks for the most specific immunodiagnostic test, boom, Western blot.
For the H in RIFIF, histopathology, imagine looking at a brain biopsy stained with hematoxylin and eosin. If you actually have to surgically remove a lesion, this is exactly what you're going to see under the microscope. Notice how an entire cysticercus is visible right there inside the bladder walls. You can literally see the extensive folding of the spiral canal and even a sucker of the scolex. It's truly fascinating.
So how do we piece together all these clues? The clinical presentation, the CT scans, the Western blots? We rely on the revised Del Bruto's diagnostic criteria from 2016. In the real world, you rarely make a call based on just one test, right? You build a solid case combining clinical, imaging, immunological, and epidemiological data, all standardized right there in Del Bruto's guidelines. Finally, Section 5, Treatment and Prevention. Let's step into the shoes of the attending physician to manage the patient in front of us and see how we prevent this from happening to others. When you're writing out your management plan on an exam, you want to bucket your treatments into 3 super clear categories. First up, antiparasitic. Your mainstays here are albendazole or praziquantel. Second is symptomatic care. And honestly, this is arguably just as critical. You absolutely must control the seizures with antiepileptics, and you've got to use high-dose glucocorticoids to manage the severe inflammatory reaction that kicks in when those antiparasitic drugs start killing off the cysts. And third, surgery. Open craniotomies aren't really a thing anymore for this, but endoscopic removal or shunting, that is absolutely indicated if the patient develops obstructive hydrocephalus. On a public health level, prevention is pretty straightforward in theory, but definitely challenging to implement globally. It all comes down to good personal hygiene to stop that autoinfection, proper fecal disposal to keep eggs out of the food and water supply, and aggressively treating humans who already have the adult tapeworm. Oh, and here's a cool fact. From a veterinary standpoint, there are experimental pig vaccines being developed right now that specifically target the Taenia solium oncosphere antigen. The goal is to just break the cycle right there at the intermediate host level. All right, we've covered the life cycle, the clinical presentation, the diagnostics, and the treatments. But before we wrap up this explainer, I want to leave you with one final thought. It's the most classic exam vignette you're ever going to encounter on this topic. If an adult patient presents with new onset epilepsy in an endemic region, what parasitic infection absolutely must be at the top of your differential? Keep that answer, neurocysticercosis, locked in your mind, and you are going to completely dominate your exams. Thanks for hanging out and learning with this explainer, and keep studying smart.